Hello and welcome back to Fiskaspjall and the second part of my chat with Abbi and Adam Smith. I'm your host Alessandra, and if you have not listened to the first part of this conversation yet, go straight back one episode and start at the very beginning, because you do not want to miss this origin story. In today's part, my guests and I talk about ethics in working with laboratory animals and explore what can happen when researchers talk shop at the dinner table. And as always, stay until the very end to catch our latest fun fish fact. Everywhere we go, there is something to see. So dive into the blue to discover the world of fish. Fiskaspjall, where we talk about all things fishy.
So you've described both how you work with these fish. Is there anything that you struggle doing to them, with them? Is there anything where you're like, I know why I do it, but it's hard for me sometimes?
So one thing that I really like about zebrafish compared to mice is that I feel like our methods of anesthesia and euthanasia are much more humane, just because it's a lot easier to anesthetize a fish or euthanize a fish than it is a rodent. And so there's nothing that I do in my current research that I feel uncomfortable with, because I feel like I am handling them in the most humane way possible. For example, zebrafish actually are not governed by any laws or regulations through the first 5 days of life. It's not until after 5 days, when they can free feed, that they're actually considered a living animal that needs any protection. So you don't need a license to work with them. You don't need a license to experiment with them. Under 5 days. But that also means that technically you don't need to provide anesthesia before you do things. But I always do. So I always euthanize them humanely or anesthetize them before I euthanize them. Because even though they're only, less than 5 days old, they're barely, a developed embryo.
They're barely a thing.
Yes, exactly. I just try to reduce any amount of, pain or suffering that they might possibly experience. So I feel pretty comfortable in my current work. But of course, reading the literature and even just experiencing, like seeing what other people have done with their model organisms, even if you do everything you can to reduce any discomfort, sometimes, I don't know, I would have trouble actually carrying out those procedures. But I'm pretty happy with how my current work goes. And, yeah.
So you mentioned that it's easier to deal with fish from anesthesia point of view, but what makes rodents so much harder to work with in that sense?
So in fish, you can pretty much always manipulate their environment. So like if you're anesthetizing them, you can just add a chemical to their water that then puts them to sleep. Whereas if you are working with rodents, you can't really do that. So you have to handle them, you have to inject them, you can use inhalant anesthesia, similar to what they give to people, but obviously you can't tell a rat or a mouse, sit here and calmly breathe this in, so they're panicking because they're in this thing. So there is always, when you're dealing with rodents, some sort of difficulty handling that you can avoid with fish. So for me, I have sort of a cognitive ladder of things I will and won't work with. So fish, I don't do anything that I consider problematic, especially right now I'm working with embryos. Salmon develop so slowly. I have in the past done a lot of behavioral work with adult fish in the cichlids and in the knife fish or the electric fish. Not an option with salmon. So we're purposely using the fact that salmon develop slowly as a way to look at embryonic development. But it also means we're not doing adult behavior. And I'm okay working with mice, don't particularly like it, but that's less from an ethical perspective and more from I don't like working with mice. And there's always the knowledge that if you work with them a long enough time, the odds are almost 100% you're going to get an allergy to them. Is a big problem with working with rodents. But I will not work with rats. So rats are too smart, too responsive to their environment. And so rats are my cutoff for things I won't work with. Anything that is a rat or smarter is off the table.
So speaking of ethics, and you do salmon research, I think this is one of the hot topics in Iceland right now.
Oh, yes.
The salmon farms. And Hola does a lot of aquaculture research as well. So we get some of the aquaculture side. We have the diploma students that usually work in the industry. So we get their input, but then you read the literature. And I find it so fascinating what then transpires in the media and what people, like everyday people, think about it.
So I try not to tell people I work with salmon specifically.
But here we are on the
we're
podcast telling the world.
I mean the general public. Like if we're talking to our neighbors, like they know generally.
Well, let's hope that the general public actually listens to this.
Well, that's
so
true.
you'll be outed.
And so what happens here in Iceland is there's a really big pushback against the salmon sea pen farming and then escape events and interbreeding. And so my work is specifically focused on understanding salmon development so that we can hopefully improve technologies for making them sterile without having off-target effects that might hurt their welfare or that might be incomplete. And so I'm very much working on the academic side, but in parallel with aquaculture goals so that, when these sea pen escapes happen, because they are going to happen, especially in Iceland where the regulations are a little lax, to be perfectly honest, that at least the interbreeding with wild populations is not possible. And this is an even bigger concern in places like Norway. So Norway is currently where a lot of salmon are shipped to, is having an immense amount of trouble with not just escape events, but also they raise salmon at such high densities in their sea pens in their fjords that it's causing low oxygen in some of the fjords. It's causing excessive algal blooms because there's so much salmon waste in the water. And so there's this litany of problems that come with different regulations in different places. And I'm hyper-focused on this one thing because the traditional method of ensuring sterility in most salmon aquaculture is inducing triploidy, so giving them an extra set of chromosomes, which is easy to do in salmon, actually. You just expose the eggs to high pressure for a certain period of time. They do not lose the set of chromosomes they normally would from the egg, and so you end up with infertile triploid fish. But those they have potential health problems in imperfect environments. And when you're raising fish in the actual outdoor world, that is inherently an imperfect environment. It fluctuates. It's reality. And so the EU has been talking about outlawing triploid salmon in EU waters for a few years now. And so eventually, those triploids won't be a viable option. And so the goal is to come up with a non-genetically modified but highly effective method of inducing sterility so that they can be raised in sort of traditional aquaculture settings without being triploids, but without having the potential for cross-contamination genetically with wild stock.
Do you think we're close to a solution, or are we still light years away?
It depends on who you ask. I'm going to go, we're still quite a ways off. Because a lot of the techniques that are currently being tried are techniques that were developed in zebrafish and have proven to be successful in some other salmonid species like rainbow trout, but those are actually pretty distantly related to Atlantic salmon, which is the specific species we're working in. And in salmon, a lot of the techniques work, but at pretty low frequency. So they work, but just not nearly as well.
So there's still some time until the EU can slice all of the triploids away, I guess.
I really have no idea what the legislative bodies are going to do, because they kind of change their decisions based on there is an increasing need for sustainable aquaculture for protein delivery for just human markets, because so many wild fisheries are collapsing. That basically, in order to meet protein demands for a growing human population, aquaculture is needed to make up that gap. But the question is, how do you do aquaculture in a way that best sort of balances ecological needs with human meat consumption needs? Because it's unlikely people in a lot of Western cultures are going to say, well, we're just going to stop eating fish.
Yeah. I mean, I come from a country that is landlocked, and we traditionally didn't eat a lot of fish. So I think I probably eat more fish now than I ever have.
Oh yeah, we absolutely do as well. You know, we both grew up in Midwest, and I mean,
there was fish, but it wasn't good.
Yeah, exactly.
I mean, for us, it was a posh thing to eat on Fridays because I grew up Catholic.
Yeah.
But that was again for the posh people. So we just ate vegetarian on Fridays. So how is it to work with someone in the similar field? Are you coming home and you're like, we are not talking about work. We have complex lives and we do talk about other things, or is it?
Talk about work.
We talk about work all the time. Yeah, we do. But it's fun for the most part. Sometimes Adam will drone on and on and on, and I sit there politely and I listen, but
and sometimes someone else talks about their tissue processor that they're very excited about. But we have not different interests, but our interests are sort of a Venn diagram. So she's more the biomedical side, I'm more the evolutionary side. And so it doesn't get stale because we have different perspectives. So we're talking about sort of common themes, but from different perspectives, and we are interested in different things. So it's not like we both read the exact same papers that day and are just discussing that paper. It's sort of a, hey, what'd you see today? Oh, that gives me a good idea. Or why are you talking about that? That's incredibly boring, which is sometimes that happens.
It's nice too. We can proofread each other's work. You can
proofread my work. I'm a terrible proofreader.
Well, yeah, that's true.
I try. I'm just not good at it.
Do a lot of brainstorming together. And yeah, it's also fun now that I'm back into fish work. He gets to, get even more excited about fish. And yeah, it's really fun. And we actually just published a paper together that basically came out of these dinner time conversations. So it got published today, actually. And so it's in evolutionary biology. And we're basically discussing in the paper the, maybe you can describe the evolution part of it, and I'll describe the zebrafish part of it.
Well, so it came out of your zebrafish work first, though. So start with that, and then we will.
Yeah. So when I was working with the smoothened heterozygous zebrafish that I used for my research, I was noticing some differences in phenotypes between the heterozygous fish and the wild-type fish.
The smoothened heterozygous?
Yes. So smoothened is the gene that my research is focused on. It is a gatekeeper for hedgehog signaling, and hedgehog signaling is a very important developmental pathway in essentially all vertebrates and even many invertebrates.
Cell signaling pathways are like a chain of dominoes. Once the first one falls, it sets off a precise sequence of reactions. But if something blocks the path or knocks the signal in the wrong direction, the whole process can go horribly wrong and lead to serious consequences. This is especially important during development when things are being built and organized for the very first time.
This hedgehog signaling pathway gets reactivated in osteoarthritis, and then also in some other cancers and diseases as well. But that's why we are focusing on this gene. And so we started talking about like how the gene dosage might affect the phenotype, and particularly I was seeing an increase in different jaw morphologies, in some other skeletal changes like hunchback and scoliosis. And I found this interesting, of course, because I'm studying this mutation that causes a joint disease, osteoarthritis, or is associated with osteoarthritis. And so Adam starts talking about, well, this probably has some evolutionary implications or evolutionary background. So he started doing some evolutionary analyses for me, and we brought this all together in this paper. And it also became an aim of my dissertation that was just kind of sort of a spur of the moment decision to just really focus in on this and make it a good story and a nice paper.
Well, congratulations.
Thank you.
So spur of the moment is an oversimplification, because this is a discussion that scientifically speaking, it was spur of the moment, but this is like 2 months of dinner conversations, because she's showing me pictures of zebrafish and telling me what she's seeing, and I'm thinking, in teleos, so bony fishes, these changes in head shape are like the kinds of things we see in species diversification or in developmental plasticity. And I was like, that kind of looks suspiciously like, how skulls have evolved in this completely different group. And then, oh, look, that body shape is kind of weird. That kind of looks like a individual of the order Siluriformes, which is what we call catfish in the U.S. And she's like, what are you talking about, Siluriformes? Oh, catfish. And so I have a background in, like, specifically how gene duplications evolve to sort of underlie adaptations or even novel traits. And so that's sort of the biggest overarching question in my background.
So gene duplication is when the whole genome gets duplicated?
So both that and duplications of individual genes. And so in teleos, we tend to think about the whole genome duplication that occurred after they split off from us. So very long ago, before coelacanths split off, shortly after they split off from all those other fish, the fish had a whole genome duplicate, and then they speciated. And so there's as many bony fish as there are all other species of vertebrates combined. About 20, well, the number keeps changing, but I think the most recent estimate is like 25,000 species of bony fish. And if you look across species, it's estimated that on average 15 to 20% of those genes that came about from that initial whole genome duplication remain in different species. And so if you have a highly dose-sensitive gene, one of the ways that evolution deals with a sudden new copy, at least what lab experiments suggest, is by removing that extra duplicate.
Can you explain what a dose-sensitive gene is?
So a dose-sensitive gene is one where if you create or remove a copy, you end up with an immediate difference in the amount of RNA and protein around, because typically each copy is going to get expressed at a similar level. So like if you double a gene, you very often immediately double the expression of that. And for a lot of genes, that's fine. The organism is like, yeah, whatever. I have twice as much, no problem. I can modify it.
So you buy twice as much, you cook twice as much, you end up with twice as much right away.
But sometimes that twice as much throws everything off. And this is particularly true of like developmental patterning genes, like smoothened. And so when you see the mutants Abbi is working with, people had said, well, this heterozygous mutants are totally normal. She's like, those don't look totally normal to me. And so that leads to the, well, if you only have one good copy and then you get deficits, otherwise maybe it's a dose-sensitive gene. And so we come up with this hypothesis that now one of the nice things about fish is they have sequenced genomes from so many different species that have been sequenced for different reasons. And then just this data is all deposited. And so we just started pulling up genomes we could find. We ended up doing, the final analysis included about 40 different species spread across the teleos tree, just digging bioinformatically through sequences that are already there, finding ones that are complete. And then we now have this big tree of very distantly related and close related species that we can say, how has this gene evolved? And in particular, does this gene retain copies or not? And so if it doesn't, there's a good chance that over evolutionary time, this gene is dose sensitive. So evolution is repeatedly removing it anytime it duplicates. And so we have sort of this in-lab stuff and this evolutionary stuff that comes from a wide range of species that sort of leads us to this one conclusion, then theoretically can be looked at in depth. Abby's advisor asked, how many years would it take to replicate this in the lab? And I said, if it goes well, only about 10,000. So
Easy.
So we're not going to be able to make a duplicate and see if it evolves out, but we can look at it using different tools from different sets of species.
Yeah. Yeah. I mean, 10,000.
Only 10,000 years. Because it's
piece of
like
cake.
20,000 well, at 20,000 generations, we might be down to like 7,000 years.
It doesn't matter.
It matters
I'm
to
not
me.
gonna do it.
I see how those dinner conversations go.
Exactly.
Exactly.
All right. I have some quick fire, like either or questions.
Okay.
Okay.
It's going to be very hard. No. Freshwater or marine?
Freshwater.
Man.
I've
Okay.
only
sorry.
ever worked with freshwater fish.
I know. I have only worked with freshwater fish, but marine fish are usually much prettier to look at and can be very, very interesting.
No, they aren't. Cichlids are amazingly pretty. They're prettier than zebrafish, yes.
Okay. If cichlid is your go-to, then.
I love them. I worked with them for many, many years.
And you still love them. Which
I still love them. They were the first fish I bred when I was 11. No, 10.
The first love.
Yes, exactly. First love.
See, my first love of fish came from when I took a marine biology course when I was in high school. And my great aunt was actually one of the instructors for the course. And my favorite teacher from high school, Mr. Patterson, he was also one of the instructors. And we actually took a field trip to Key Largo in Florida. And we went snorkeling every day, multiple times every day, and got to identify fish and, well, all the other creatures down there too. It wasn't just fish, but that's sort of where my first real love of fish came. So I guess it's marine for me.
Next, model or non-model organism?
It depends on how you define model, but I would say non-model.
Whatever your definition.
So my definition of model organism would be include using like cichlids as a model for visual evolution because they're good for asking specifically that question. I like to try to take species that are a model for one thing and ask a different question with them. So it's kind of like non-model research. And I just feel like a lot of times it's more fun to be like, hey, no one's looked at this problem at all. Can we do that? And sometimes it works and sometimes it doesn't.
I think I would go with model because I'm really interested in developing like a zebrafish model organism community here and getting more people excited about using them.
Then I guess fieldwork versus lab work. I mean, like fieldwork as in like working with the actual animal and lab work in the actual lab pipetting, I guess.
Field work. I love working with the fish.
So I would say field work, except now that I'm at so in the U.S. I'd be called a principal investigator, PI. Here they call them group leaders very often. So largely it involves telling my students what I think is a cool idea and then they go and do it. So I like it when they go and do field work, but I spend a lot of my time writing and grants and stuff like that. So as usual, as you go farther in your scientific career, you spend more time at your computer and less time
Doing the fun things.
doing the fun things.
But you would like to do the fun things.
Oh, yeah.
Yeah.
I still get to do them sometimes.
Then coffee or tea?
Coffee.
Coffee.
But I do drink tea as well, but coffee is my daily necessity.
I drink. People are even like, you're in the coffee room a lot. I'm like, yes, but I just fill my mug and leave. It's not like I stay there.
Summer or winter?
Ooh,
That's a harder question here.
it is a harder question here.
Winter.
Well, there
Yeah,
are
I was-
only the 2 seasons.
Yes,
Exactly.
exactly. Yeah, I would say winter overall as well. Back in the U.S., definitely winter. It got too hot in the summer. So I have adapted really well to this climate. But still,
Back in the U.S., I say neither summer or winter. Spring or fall, much better.
yes.
Here, it's definitely winter because it's actually unexpectedly harder to adjust to the long days than to the short days.
I agree.
Because we already work in labs without light.
Yeah,
exactly. It's our
I'm like,
natural environment.
I'm totally used to this. I live in my tiny sort of aquarium that is my office.
Big international conference or small national conference?
Small national for me.
I go big international. Yeah. I like seeing so many different techniques being utilized that we don't necessarily have immediate access to here. Of course, we can collaborate with EMBL and throughout Europe and stuff so that we can have access to these different techniques. But it's that added, hurdle of then you have to ship your samples, you have to go there, you have to get your travel costs funded and all this. So I don't know. I like the big international conferences. I like seeing what's out there that we don't get to see on a daily basis here.
And so she actually exactly hit on why I like the small national ones, because you develop lots of really cool ideas and collaborations at bigger national conferences, but they're so hard to actually institute because there's so many logistics, especially being here in Iceland. Whereas at the small national ones, it's much easier to come up with collaborations that you can actually execute. And largely the Icelandic research community is a lot of really nice people that are open to collaboration. And so it's fun to sit down and talk with them and then come up with ideas that it's like, oh, we can start this.
It's like doing Tinder on holidays versus doing Tinder when you're actually at home.
Yeah, basically.
So what is your least favorite part of being a scientist?
Grant writing.
Yeah, I would say same thing. Chasing the money. That's, it's just not that rewarding. I'd rather do the research, and even writing papers is so much more fun than writing grants.
Yeah. And what is your favorite part about being a scientist?
Teaching. Easy. So teaching is and so that's actually why I like being a PI or group leader, is actually training the students. That's the best part, because students, the way I put it is, they don't know what they don't know yet. So they come in with big ideas and new ideas. And it's like, you know what? That's a really cool idea that I never thought of. We should try that. And that's something where after you've been working with the same model system for a long time, you tend to get sort of blinders on to things outside of your purview or new techniques. It's easy to be like, I know how this is going to work. I know this is a good question. And students like, we should try this. It's like, that idea is borderline insane, but we're going to try it. And without students, that wouldn't happen nearly as often.
That's how I didn't realize that my experiment was absolutely insane.
Uh-huh.
So I just went for it.
Exactly.
Yeah, I also like training students, but more so in the technical side of things, like teaching them how to do the actual lab work and work with the zebrafish too. Adam really enjoys teaching lectures and classes and stuff as well. I enjoy it, but in much smaller doses than Adam does. But I also really like the innovation of my job, like especially being here in Iceland, where it can be very expensive or difficult to get even, somewhat basic materials or equipment and stuff. So you have to just kind of work it out yourself. And that's I think one of my strengths is being resourceful and like figuring out how to make this work with the material that I have. And I actually find it really rewarding and really enjoy that process, even though it can be really frustrating.
We were just setting up, my graduate student Gunhiller just started a new, sort of a pilot project, but it's got an overarching theme. But if it works out, it's going to feed into one of her aims. If it doesn't, it's just a neat little short-term study. And we were getting ready for it, and the day before we were getting the eggs, so we have to be prepared for when the salmon eggs come in. We are like, oh man, this is something we forgot. Oh man, this is something we forgot. How do we get this together? And she goes, I didn't know science was this much improvisation. She's like, I thought we planned everything. I'm like, we do plan everything. And then we realized what we forgot.
And that's why I say this is a highly creative process.
Yes. It's a bit like arts and crafts sometimes. And I like arts and crafts.
I call it arts and I had them making the egg baskets. And I'm like, this is the arts and crafts parts of science. And they're like, they looked at me like I was crazy. I'm like, no, seriously, making stuff you realize you need.
I
is
sniffed
a
a
huge
lot
part
of
of it.
glue preparing for my experiment. So is there anything you would like people to know, like that one thing, or is there a misconception that you would like to clear up? Big one, I know. It's so hard.
So, I mean, it depends so much on working with a lot of students. It depends. I've come to realize it depends massively on like each individual. But the main thing I try to tell a lot of young scientists, and by that I mean like early grad students, think we know far more than we do. Like they think they're going to come in, and I've had a set, especially like third-year bachelor students here, say, all the interesting questions have been answered, right? It's like, no, we don't know anything about anything. They're like, how do I come up with a good question? I'm like, just think of something. I guarantee you we know far less than you think we do. And that's the largest misconception. And I think that's a general public misconception too, is the general public thinks scientists know much more than we do, and that science is a list of established facts, not a process for learning new things. Once you've been in science for a long time, you realize that science is dynamic and changing, and it's not a list of established facts. Yes, when you're in primary school, we teach you things that we have validated with decades of experiments. But things we're working on now are not, like, it's not something we know. It's something that we hypothesize and we update and we hypothesize and we update. And it's very dynamic and sometimes daunting how many things we don't know.
Oh, yeah. Every question that we answer raises just 10 new ones.
Yes.
But I think that's what makes science fun, too. But, the public misconception of this is a very real problem, too, because you have people who honestly believe that we have a cure for cancer. And that has so many misconceptions because, first of all, they assume that all cancer is the same and that we can cure every single one of them with one treatment. And then they also are suggesting that, we have this information and we're just keeping it to ourselves because otherwise we'd be out of a job or something like that. And so
I wish.
Yes, I know, exactly.
Yeah, it's like, trust me, if I had a cure for cancer, you would know.
Yep, exactly.
And you would be able to see it in my giant house. I wouldn't live in a cellar under
the island that you own.
Yes.
Yes.
So they might not be able to see it.
Yeah, well, that's true. I would be very far away from everyone, have my own helicopter. No, I care about climate change, so.
I was just going to say how eco-friendly.
Yeah. But yeah, I think that's what makes science so much fun is there is a lot that we don't know. And I think that one of the greatest attributes of a successful scientist is being able to recognize the questions that haven't been answered yet. And I know that that's something that I'm not necessarily the best at because I definitely have these moments where I feel like I don't know where to go from here. Like, we've solved this problem. What's next? Whereas Adam is the opposite of me, and he always has 1,000 other questions.
I'm great
just
with
right there.
anybody else's project.
Yeah, exactly.
Except mine.
Well, so I'm really good at coming up with identifying questions, but not necessarily how to answer them. Abbi is really good at thinking very mechanistically about, oh, this is a question we have. This is how we can answer it. These are the things we need to think about. So we actually play off of each other. This is where a lot of dinner conversations go. I'm like, okay, this is a cool question that I have, but how do we address it? And she's like, oh, you could do this and this and this. I'm like,
And
absolutely.
this is where somebody could think you are a power couple.
Yes.
Yes.
So I want to thank you so much for taking the time to sit down with me and to do this. And also congratulations on your thesis. This must be like the day of the days. You have your thesis submitted. The paper is published,
And we get to talk with you. It's been great.
Very fun.
Thank you. So yeah, thank you so much. And I definitely hope to hear more from you guys in the future.
Yeah, this has been
It was
really
a
fun.
blast.
Yeah.
Thank you all so much for listening today. Big shout out to my colleagues at Icefish Research, Like, Teresa, Michelle, and Gudbjörg. Thanks to ASIP for handling all the paperwork, and thanks to Háskóli Íslands for awarding us their science outreach grant that makes this podcast possible. Thanks to Sandra Rybicki for designing our beautiful logo, and special thanks to Kaure for trusting me with his incredibly cool recording equipment that allows me to go and record people everywhere. And our title song was written and recorded by me, your host, Alessandra. If you also want to support us, you can subscribe to our podcast on Patreon. There you also get to submit questions and suggestions for future episodes, because yes, there will be a second season. You can also leave us a review, and please tell all of your friends and family about this podcast. It really helps us grow. Follow us at Icefish Research on Facebook, Instagram, and Blue Sky, and visit our website at icefishresearch.com. Today's fish fact revolves around Adam's first love, the cichlids. Many of them are what we call mouth brooders, which means that they incubate their eggs in the mouth. Dominant males establish breeding territories, which the females enter when they are ready to spawn. After a female lays her eggs and the male fertilizes them, she picks them back up and keeps them safely in her mouth, where they stay until they hatch and are ready to fend for themselves. Once they are ready, they still seek shelter in their mother's mouth for a day or two, but quickly leave, and the mother is relieved of her mouthful of responsibilities. See you next week with some more fishy facts.